the second webinar hosted by the International Legume Society. And today we are going to listen from Dr. Petr Smykal from the Department of Botany in Palacky University, Czech Republic. This is the second webinar we had. Last time, Christian Wig from IRAE had a very interesting webinar about the role of legumes as a source both of protein, and he talked extensively about the nitrogen cycle and put legumes in perspective of the current and future state of agriculture. Now, I guess that with this seminar, we take a step back. And today we are going to talk about domestication. Legumes have been domesticated since very long time. And Peter is going to talk to us about how and domestication of legumes is intertwined somehow with even human history. So without further ado, thanks, Peter, and we are glad to hear from you.
All right. Okay, let's try to share. So do you see my shared screen? Is it okay?
Yes, we can see.
Okay, great. So first of all, thanks again for Paolo to putting these webinars in place. I think it's a great idea to do this. It's just to meet at this in this way from time to time. So what I would like to talk today is, as mentioned, to take you a little bit back, or not little bit, but let's say some couple of thousand years back in history, and to show in some examples what we know today about the changes which happened. Before doing so, I would like to show just 2 slides where I come from. So, as mentioned, I'm based in Palacky University, which is the second oldest in our country, after the Charles in Prague. We will celebrate actually 450 years next year. And since this year, it's 200 years anniversary of Mendel, which I will dedicate my talk on the end. Actually, it's not that well known. He studied here for 3 years. Actually, that was the only university which he studied successfully, but just to let you know. So how it looks like, we are in a campus on a periphery of the town, but it's a quite good place to be. There is some land around. Actually, we are in a very fertile part of our country, very good for barley and sugar beet. And within the campus, there is also Academy of Sciences, where among others, Jaroslav Doležel, I think many of you know him, is located. So person which basically make the cytogenomics and the sorting of chromosomal based genomics. So that's based here in this campus. And there is a gene bank as well, which is I think it's good backup. But of course, we are historical buildings as well. So back to the topic. What I would like to tell you today is to first let's say, get you a little bit more flavor on legumes, just to show up how large plant family it is. And then, of course, we will zoom into the crops, particularly selected ones, which we are which we will later on, we will talk about. Necessarily, I have to touch the aspect what happened during domestication, which can be viewed as a kind of accelerated evolution. And I will later on focus particularly on 2 traits which are crucial: seed dormancy and pod dehiscence, something which actually in my lab we are studying for the last couple of years. Okay, so as mentioned, actually botanically, if you look at the legumes or the Fabaceae, are 3rd largest plant family after orchids and grasses and Asteraceae. Well, there are some which in the same position. You can see we have roughly about 23,000 species altogether. And they come from tropics to Arctic, from, let's say, annual to very big trees. So they're quite widespread. But actually what human is using is kind of small subset on the very end. I'm aware that you cannot read it properly now, but this is taken from this kind of Bible book on legumes from William Lewis, which is very nice book. I recommend it highly. It's hard to buy it nowadays, but it's great. And we will talk specifically in a annual crops, grain legumes. Why the legumes are called legumes? It's comes because of the pod shape, which is the botanical name legume. Some has a typical structure of the flower, which looks like a butterfly. That's why the one subfamily is being called like Papilionidae. I think this you are familiar with. Most, but not all of them, are able to fix the nitrogen from air, but this is not the topic of today. I'm sure that we will touch this aspect. It's a very important one. And yeah, we will go gradually in a domesticated. Actually, this is perhaps you can take it as advertisement, but we have done some years ago, I think, quite nice and useful review trying to show up how the phylogeny and the genetic diversity of legume crops, let's say, can be used in combination of botany and plant breeding in this way. So what I will talk today, I will not touch about any trees, but there are many of the legumes which are quite interesting in terms of the timber or let's say the dyes or the source of very specific like Arabic gum or even the insecticides, for example. But today we will mostly focus on one particular subfamily, which is called Papilionidae, and as labeled in a red, there will be some species which we will, let's say, talk more. Particularly, we will talk more about the chickpea, lentil, pea, which are closely related, including Vicia faba. And as you will see, the most genetic studies has been done on soybean, particularly, I think, because it's of economical importance. And from the same clade is the Phaseolus, the common bean, coming from. These are not the very updated numbers. I cannot find the better ones, but arguably we can say definitely the forage and grain legumes are quite important economically, as they have been grown in quite some surface on let's say cultivated land, and they contribute perhaps about 1/3 of the primary crop production. As I said, the numbers might change today, but definitely they're very important nitrogen, or let's say the protein nitrogen needs for human as well as animals. And as mentioned, they're also important in a term of the fixation. I think this aspect will or is becoming quite important nowadays in many of the scenarios. I apologize for the Czech, but what I want to say in this picture is just to remind you that the crops which we are relying on using globally, they come from very different geographical regions. Well, these regions has been established by Nikolai Vavilov back in 20s of the last century, and they're basically holding through. So one which we will focus today mostly, it's coming from, let's say, Mediterranean region. And because of the common bean, we will go a little bit in America. And because of soybean, we will go in Asia. But as you can see, they come from various parts of the world. If we go specifically for legumes, and this is not the extensive or complete list, you see they also come from Asia, Africa, Europe, America. So it's geographically widespread. And I will emphasize once again this point on the very end, because I think it's exciting. Once we know the specific domestication related genes, it will be very interesting to see if the same pathways, and this is what I will show you in some examples today, has been altered independently, because they have to be independent. You know, they come from different regions, they are come from different genera, so they have been independently domesticated by humans. And this is quite exciting, I think, in terms of the evolutionary perspective. Still being in an introductory part, it's also quite good to remind us that typically it's like a partnership between the cereals, which of course provides mostly the carbohydrates or starch, if you like, or energy, and legumes in any of these parts of the world, because the legumes has been a source of proteins because of the seed composition. Basically, there is no endosperm, and the entire seed is embryo. So largely, there is a good proportion of proteins, and that's why they are so important for human diet. As mentioned, of course, we know by archaeological excavations from various regions, and some are, let's say, better sampled because of the climatic properties, typically like Middle East, if you like, the current Turkey or Syria, though let's say the Mesopotamia region or Fertile Crescent. We know that the legumes has been taken by humans a long time ago, together with the cereals. What is shown in the graph, it's a study published in PNAS back in 2014, which shows in these numbers are in 1,000 of years before present, and that just illustrate the frequency of archaeological finds for wheat, barley, lentil, pea, chickpea, and flax, for example. And as you can see, we have quite good evidence that they have been, let's say, harvested. And that's a question if they have been harvested from wild, like being foraged, or if they have been already cultivated back in like 12,000 years ago. And I will come back to this aspect again. And of course, there are crops which are not so old. And I will give you example in a minute in a lupin, which is one of the recent domesticated. There are some intriguing crops. For example, faba bean, which is, let's say, has been cultivated in Europe for quite a while, but it's recently is not that much. But it's interesting, we don't know the progenitor, because there's some, let's say, I don't want to go in very detail, but all the current species which are growing in Mediterranean, which even looks like a faba bean, they are not compatible with the faba, because there is a one chromosome difference, which makes them basically reproductively isolated. So there is a kind of enigma. What was the progenitor of faba bean? And this work basically illustrates that we have some source which eventually could be studied, maybe one day, because there is a technique improvement. Maybe we will be able to see some DNA analysis from such a kind of charred seeds. Of course, this is not a perfect material, not like a bones in animals, but hopefully one day we might be able to answer some questions in terms of if these seeds has been already coming from cultivated crop or harvested progenitor. But this is one of the another example which we did together with our good friend, which is unfortunately not with us anymore, with Alexander Mikic. We tried to get some hands on DNA from the Iron Age pea, which is not that old, of course, it's 1,500 years. At that time, we didn't have that much, let's say, genes to look at it, but we simply ask if this pea was like colored, so if it was pigmented, because the seeds has been, let's say, not preserved in terms of the color. And by looking the chloroplast DNA at that time, we look at if this relationship, if it's like cultivated or if it was like completely wild. So I think this will be better in the future, or maybe even nowadays. I am not aware of any other study published. As I mentioned, there's some, there is still a potential. I will show on the very end. I think we still can domesticate the new crops, including legumes. And one of the examples, which is quite recent, is narrow leaf lupin, or including the Lupinus luteus, the yellow lupin. But there are other lupins like Lupinus albus, which has been domesticated back in Egypt time, or ancient Egypt. But this particular two has been domesticated very recently, essentially like 200 or 150 years ago. And actually, this is a great job which was done by Australian colleagues, which took the lupin and essentially in course of few dozen years, and this is shown in a panel in the middle, what is shows that basically like removal of a vernalization and photoperiodic requirement in a breeding perspective in a course of like 40 years. So basically the very original, slightly domesticated, let's say, accessions has been flowering considerably late because they have a typical Mediterranean, let's say, phenology, which has to be altered. Basically, it was like necessity to turn it into the annual crop. And this also showed the perspective for the future, I think, that we can still do something more. I will show you on the very end. If we say that something was selected, basically, this is holding true for all crops, including, of course, domesticated animals. We took the subset of the diversity which exists or existed. And that's essentially leads to the bottleneck, which is illustrated in, this is not the newest study, but essentially all studies, if they look at the genetic diversity of the crop compared to the progenitor, in this case of wild pea. So these green dots are, let's say, the accessions coming from specifically Johninus germplasm. And you see there is a still reasonably diversity, but mostly it's something in a center. So to say they're quite closely related. Compared to the wild, which are like yellow or red dots, which are unfortunately much less preserved in germplasm. We have typically very few progenitors in a gene banks. And this is one of the, let's say, message or problems for future, or even today. But they're phylogenetic, or let's say they're distant. They're definitely different. So I think they might have quite some interesting traits, which eventually could be good for drought or any climate change, let's say, arguments scenarios. Now to the domestication, before we go really in examples. So what the domestication means, essentially in a plants, and I stay with plants only, any plant species has to be adapted to the environment where it has been evolving. Let us say the diversity and the relationship was shaped by the environment, which is depicted on the very upper left picture. While later on the human has stepped into and started to, let's say, make maybe the clearance, basically open the space. Maybe it was not like making cultivating land, but basically cleaning maybe the trees or shrubs first, making the like savanna type environment. Later on, of course, the human has been involved much more, basically, has even shifted geographically crops to the new climatic environment. So basically start to be a major evolutionary force. Today, we are somewhere in this, let's say, scenario where we have the typically kind of monoculture crops in a large scale. And what will be next? That's a question. So that's I leave for the others. So just think about the influence of the environment and influence of on the human. If you look at what happened, so we have a progenitor, in this case illustrated by the maize or corn in a bottom part. And you see there is like overlap between the wild and cultivated. They are kind of connected initially. So basically, human has selected something which was, let's say, interesting, or maybe it was a little bit different plants, and started to grow it, basically. It was still not domesticated, but there was no barrier in terms of the gene flow. Over the time, of course, this kind of genetic distance become greater, but there was still no kind of barrier. So that was a time when we, let's say, started to select favorable, like non-dormant seeds, for example, non-shattering pods, which I will come to that, and let's say the traits which are essential for the kind of harvest to have something as a yield. Over the time of millennia, of course, we are in a stage that we basically deliberately cross only the cultivated ones. We disconnected this relationship unless we want something specific from the progenitor. And this I will touch again on the very end. So we are basically in a stage of deliberate breeding where we took just the selected lines which we like. They're already fully domesticated with traits just we want to have. Well, this issue of domestication has been kind of intriguing for many persons, including Charles Darwin, just to highlight. Actually, this is the first chapter of his famous book. And as you might know, he wrote another book concerning the domesticated animals and plants. And just the arguments he was putting in a chapter was saying that they're quite good system to study, but he was also aware that these crops are even today kind of not very acceptable for typical, let's say, botanist or what we call naturalist, because we still consider them something strange, you know, that it's like something different. They are different for sure, but let's say for the purpose which Darwin wanted was a very good system because of the variation available, and that's actually the model that's which of course Mendel used later on for the genetic studies. Coming more into the domestication, if we look at, let's say, through the plant families, so from monocots to dicots, from let's say strawberries to kohlrabi, tomatoes, or whatever you can think of, there is a kind of set of traits which has been altered. Some are, let's say, more specific to one family than to other. But if you look at globally, we typically, let's say, remove the toxic parts or preferably selected for less toxic genotypes. Over the time when the human has, let's say, shifted geographically, so one of the aspects was also altered was a requirement of photoperiodic requirements, for example. So having in mind, for example, if you think about the phaseolus or soybean coming from subtropical or near equator regions, they are typically short day plants. And the problem which we are still struggling is if we cultivate them way up in Canada, for example, or Scandinavia, they tend to flower late in a season, which is a problem. So of course, this has to be changed. But what are two very important traits which has been altered through all the families is a dispersal of the seeds and the seed dormancy. So typically, they're like combination of the traits. So it's not like one single trait for the given species, but it's like several ones. This is actually this domestication syndrome as a word or as a term was given basically at the same time by Jack Harlan and Karl Hammer. That's why it's acknowledged here. The question is, of course, also it's something. How many times in a given crop domestication happened? I think we will never be really able to answer this question completely, because you have to keep in mind that similar to like evolution or phylogeny, we are studying something which is currently surviving, but there will be surely some lines which didn't make it through all these millennia. And also you have to keep in mind that in many cases, they likely was like perhaps several domestications events in the given crop, but later on they come together and basically we cannot distinguish genetically if they have been domesticated several times or more. But there are a few cases which we have quite interestingly good evidence. One of them is a common bean, the Phaseolus, which is on the right bottom part. We are quite sure that there must be at least two domestication events independently. One is from the Central America, so currently like Mexico, Guatemala and so, and the other is from South America, so to say Andean center. I will touch it once again when I will have a genes. But typically in most of the crops, and not just legumes, it looks like in this Cajanus pigeon pea case. So we have like, when we do the diversity and we do the sequencing, we typically see the progenitor, which is like coming out. And then we have the big clump of the domesticated, which are like having one single cluster genetically, which suggests, or the conclusion is like one domestication event. In RP, by the way, there is quite good evidence that it must be domesticated at least twice, because we have a common pea, which is Pisum sativum, and then we have the Ethiopian pea, which is still cultivated pea, but it's always been any analysis, and there is a recently published genomic paper which show up really the Ethiopian pea is like distinct from the common pea. So they might be at least two. There are also species which are quite interesting in terms of that there are like several species from the one given genera, which is quite good in terms of the combination or comparison. So for example, in Phaseolus, I think this is a favorite species for Roberto Papa or Paul Gapps, for example, from US Davis. So we have a common bean, but then we have four other species which has been independently domesticated, which are, let's say, less cultivated, but they're still underwent the domestication. And this is exciting in terms, if we have a genes which are responsible for any of these mentioned domestication trait, to compare them across such a kind of related species, to ask the question, and this hasn't been done yet, to ask the question if the same genes or the same pathway has been altered. Well, I skipped this one. It's just, let's say, showing that definitely common bean is coming from two places. And I touched a little bit aspect that how the analysis typically might look like. this is showing the studies on soybean, which show that the progenitor, which is cursively, it's called Glycine soya, and cultivated soybean is a Glycine max, has been, let's say, phylogenetically spread definitely before the human. So the progenitor of currently cultivated soybean split like 200,000 years ago, which must be well before the human has touched it. But the domestication, which we have evidence also from the archaeological founds and also from some genetical studies, has been around like 9,000 years or 10,000 years ago. And what all these trees shows that there was very likely for some time the gene flow between the progenitor and the current varieties. So they have not been like exclusively, let's say, being separated. This also shows that some lines might be like not surviving till today. And for example, that they show also the American, which are the, let's say, the very elite varieties, are kind of very narrow subset of the soybean. So just keep in mind this kind of scenario. Now more zooming into these mentioned two traits. So this is illustrating seed dormancy. There are various types of the seed dormancy. What is mostly studied is like cereals or Arabidopsis model, which is about the interplay of the phytohormones like gibberellin acid and abscisic acid, which is on the right. In legumes, and not just legumes, but other species too, but legumes is quite prominent. There is barrier in terms of the water permeability. So basically the seed coat is called kind of very hard, and it doesn't imbibe, doesn't take the water into, unless it's, for example, scarified, which is shown in this case. And you see, once you scratch the seeds, they start to imbibe and germinate. So this is something which we are interested. How it would look like if we would still cultivate some dormant seeds of pea, but it will be the same for any legume essentially, is shown on the upper part. It's experiment from Shah Labo done in Israel, which show from the left upper, there is the same plot with the same amount of the seeds, and on the left upper part is a wild pea, in the middle is like landrace, or let's say primitive domesticate, and in the upper right is the modern crop. And the seeds has been left just for the first rain to come, and you see the result. Basically, you see the crop has emerged and is growing, while the wild is just like single plant perhaps. The rest are still alive, but they simply wait for the another rain or another year. And this is something which you don't want to have, because you will not be able to harvest enough seeds. The other argument is, of course, we know that all legumes need to be soaked to basically appreciate or to have it nutritionally good. So that might be the second selection. So we have done this in, let's say, this is our experimental model. So we are working with the domesticated and wild peas. We still can cross them. They come from, let's say, different color pigments and so on. This is just to illustrate how the seed coat looks like, and this is the same for any legume, if you like. So there is a thick layer of so-called macrosclerate, and there is a part which is called, looks like a bones, that's why it's called osteosclerates, and there is the parenchyma, which is touching the embryo inside. And what we are talking about is the thickness. We know that the wild are much thicker and the cultivated are much thinner. And this holds true for any legumes if you do comparatively. If you compare soybean, common bean, peanut, they will looks like the same. We know by genetics that we can decouple this. We might decouple. It's not about the sickness. It is about some chemistry components, which is illustrated here. So this is a dye of the Schiff staining, which this pink color illustrate where the water goes. So on the right, you see the cultivated pea, or let's say the cultivated legume, and you see the water goes quite quickly and the cells are kind of hydrated very rapidly. Compared to, or contrast to the wild ones, where you see the upper part only gets hydrated and there is like a barrier. It's not a distinct cell. It is still the same cell. It's one single cell layer. But there is some called like linea lucida or light line because it's visible on microscope. And this is preventing the water entry. So there is something happening there. We have done, basically this has been done in other scenarios, which I will show you in a minute. So you can still cross the cultivated with a wild progenitor. There is no reproductive barrier typically. You get a fertile progeny and you can screen for, right? So you basically testing the imbibition rate, eventually germination, which means the root coming through the seed coat. And so the cultivated gets rapidly hydrated, rapidly germinate, while the wild one is doing this very, very slowly. We know by, let's say, some years before, by classical genetics, in these three, which we are currently interested, lentil, chickpea, and pea, that there are likely two genes or two loci responsible for seed dormancy, and one for the dehiscence of the pot. Where we are now, basically, we know the position by, let's say, chromosomes, but what is a typical, of course, scenario that it ended up in some region, which is still large because of the size of the, let's say, the genome of all these crops is pretty large. And then you ask which gene it is in the region. I should also mention we are still, I think what is needed is to have the genome of the progenitor, or whatever you call it, like pangenome, because typically we have like single genotype being sequenced, and they might be a case that we might be looking for the genes which are maybe even not present in a typical domesticate, but that I will mention on the very end. You always ask if they're synthetic or not. So maybe in a lentil and pea, there is some synthetin in a pod dehiscence. But now I will take you to the work which has been done by others. So I have selected some examples which has been studied in let's say, which has been successfully identifying the gene. There are not many, don't worry. I will show you just few which are selected. So this is a scenario which has been done in soybean, taking the wild soybean progenitor and the cultivated soybean, crossing them and looking through the progeny. And looking about the imbibition, that's illustrated on the B on the left. So again, the seeds are either imbibing rapidly or they basically don't do anything for a long time. By, let's say, doing classical mapping, they have been able to come to the candidate gene in this sense. So they identify the region on the chromosome 2 of the soybean. This is work from 2015, I think, and they come to the gene. So they identified it encodes something called calcineurin. So let's say the protein which is involved in some aspect of the calcium, let's say the transport, and they show that there is a difference in the allelic composition and the expression pattern. they have been able to complement it, by the way. So they basically did some transgenics, and they could show up that this is very likely indeed the gene which is responsible. They have been able to identify the specific, let's say, point mutation which is responsible for. What is interesting that very same time, very same year, there was another study showing like different gene in a soybean responsible for the same trait. And I will show you in a minute what could be a reason. So in this study was like showing that this is gene doing with a like, we know the glucanases are about the let's say rigidity or flexibility of the cell wall, which makes sense, basically something which might be like relaxing the connection between the cells. I will show you in a minute in a model. So they have identified this gene, and there was not like unfunctional gene, it was like a change which led to the amino acid change, which is indicated in the bottom. But what is the message? Actually, it's quite, sorry for the jump, but I will show you hopefully in the next slide. There was interesting relationship in terms of the environment. So sorry for change, but now we change from soybean to phylogenetically related, but geographically very distant species like a common bean, which is an American one, right? This is a study which actually it's interesting because they haven't compared wild and cultivated, but they have compared two distinct cultivated genotypes, and they look at the speed of imbibition. And hopefully what you can see in this picture is like tomography scan, which illustrate the upper genotype is imbibing more rapidly in terms of the 3 hours. You see this arrow is showing the water entry point, because in Phaseolus it's a little bit different than what is in P. There is like specific entry point in a land where the water goes through, and then it's spread all around the seed coat, compared to another genotype which is doing this much slower. It imbibes. It's not a wild, but it imbibes considerably later. So by doing classical genetic studies, they come to the region. Then they look at the expression pattern of the genes which are encoded in the region. And what is illustrated on the upper part is just like two genes which are differentially expressed. And particularly one was very dramatically expressed differentially between these rapidly and slowly imbibing genotypes. And by looking at the homology, they identified that it encodes the pectin acetylesterase. You know, the pectins are quite interesting compounds because pectins are kind of jelly. Basically, if you make a jams or something, you may add some pectins. So they are known to absorb quite a bit of water. And the modification of pectins are known to be responsible for alteration of the rigidity of the cell wall. So what they have found, that when they look at the wild genotypes in blue in this map, they show that they have a, let's say, functional allele, while the cultivated ones, they have mostly, not all exclusively, but mostly non-functional alleles because of the insertion, which leads to the frame shift. And this is just like, let's say, the illustration. On the left, what you see, there is like what the proposed model, the high activity of pectin acetyl esterase together with the calcium ions make very rigid connections of these, or in the middle lamella between the cells, while in the, let's say, downregulation of pectin acetyl esterase leads to the kind of relaxation and the loose, let's say, the loose cell wall. And the picture which is shown, it comes from our work on P. And this in pink, this is a kind of paradermal section. So it's not a cross section, but it's like a horizontal to the surface. And in a pink, there is a specific staining for the pectins in the seed coat of the P. And you see there is a lot of pectin matter staining. And we know the pectins are definitely between the lamella. They're kind of glue of the cells, together with, of course, some cellulose or hemicellulose, which are much more complex. And it has been shown in some other seeds, or not only seeds, that actually the modification of pectins is quite responsible for relaxation. This is maybe provocative, but I would like to mention one aspect which is kind of intriguing, and not just in legumes. It's intriguing if you look at globally on the crops compared to progenitors. Typically, crops are far less pigmented. And there is a question why. Was it because there was a kind of flavor, or was it really something so important? Maybe there was some religious aspect, or maybe simply they have been so distinct that it was just selected for. And as I said, this is not just for legumes. You can see it across the crops. This is just showing that actually we know, even by Mendel work, that there is a transcription factor which you can find homologous genes across all these related legumes. And there is a question if there are some genes nearby which has been like swap selected, perhaps. And that's, as I said, it may be something to discuss. But if we know that, of course, not only unpigmented, we have variety of seed coats which are still cultivated, which are still edible, and we actually even like them. So there are many different, let's say, cultural scenarios where, so it's not only about the seed coat removal of the pigmentation. This is last promotion of our very recent paper, which was quite interesting. Actually, by looking for the dormancy genes, we came across interesting gene which is not responsible for dormancy per se, but which encodes the polyphenol oxidase, the gene involved in oxidation and polymerization of phenolic compounds, which definitely has been selected across the domestication, because we found high preference for one single allele in most of the domesticated peas, and as a consequence, there is no hilum pigmentation. So basically the hilum is pale. It's interesting. It might be a kind of defensive reaction, and also it's something which we are still studying. We found it recently is not only in case of pea, but it has been also now shown in faba beans, and I suspect it might be true for many other legumes, actually. Touching the point of if there is the trait which has been, it's a question also always, if the domestication trait has been selected like after the cultivation, or was it selected already from something which existed before? It's quite likely that our kind of ancestors has already chosen genotypes which has been, in this case, less dormant. We look at the let's say wide range of the wild peas, and not all of them are dormant. There are some which are still wild, but which are considerably less dormant. And this has been shown also in terms of the cereals, that likely our ancestors has selected for the variants which has been present in natural populations, maybe in a low frequency, but they have basically started to work with something, let's say, more useful. So we show that there is a relationship to the environment, not surprisingly, but that's another story. How I am about time? Do I have still 5 more minutes or something like that?
Yeah, sure.
Yeah, sure. Okay, so this is, let's say, now coming to the second trait, which I mentioned, the seed dormancy and seed dispersal. And again, the legumes are fortunately lagging behind, but the situation is getting better. I will show you now. Because far better studied are unfortunately still economically important cereals. But so what is about the pot dehiscence? This is showing the typical scenario. So in any of the legume crops which we grow, we don't like to free shatter seeds, because otherwise, once a legume is ripe and mature, they will just ballistically basically protrude and shatter the seeds. And this is our work on a pea, but I will show you there is the same scenario true for common beans and soybean. It's about the lignification. So what you see here is a cross section of the very upper part, so to say suture, so the part connecting these two carpel. And hopefully you can see there is a distinction in this pink colored region, which is connecting two halves. So you see in wild, there is like much, let's say, more violet region compared to the cultivated one, where is a disconnection. And this staining is for lignins. So now we will be about lignins. So the very first study which was done for the same trait was done in soybean again, comparing and genetically crossing the cultivated soybean with a wild soybean and looking the dehiscence. They come to the region, they have like several candidate genes in mind. And what is showing here is a expression pattern. And what you can see, there is a one straight, very different gene, which is differentially expressed between these two, so the wild and cultivated. You see there is a big difference in these two. And on the upper part, there is shown where the protein of the given gene is located. This green color is an antibody showing the localization in the connection in the pot. So what they have found, that there is a mutation in the regulatory region, so basically in a promoter part, and that's why there is a difference in expression. As a consequence, there is a difference in the thickness and lignification of the connected tissue in the pot. So that was one gene. But interestingly, another group has identified another gene responsible for the very same trait. And this gene is quite known because it's called dirigent. It's about the lignification again. So the same scenario. There is a mutation which leads to like a premature stop codon, which is the shattering resistant or shattering susceptible alleles, and they differ. But what is interesting, just think about where the soybean come from. Soybean come from the southeast or South Asia, which is relatively humid environment. And as I said, there have been two genes identified from two of these studies, which I showed before. So one is on chromosome 7, another is on chromosome 16, and they have been quite clear signals in association mapping studies. And when they look at geographically and try to relate the relative humidity pattern, so it was quite interesting to see that actually this is initially it was not the essential gene for the domestication of the soybean, because soybean was domesticated in relatively humid environment where was no problem with the shattering. So basically the pod was relatively quite keeping together. But later on, as it moved to the drier environment, and then later on, of course, particularly to North America, there was preferentially selection for the shattering resistant allele, which was necessary to keep the seeds, of course, enclosed. Coming to the end, or let's say comparable studies was done very nicely on soybean. And what is intriguing, as I mentioned, is always to compare independently domesticated species. So Mexico is a different environment. It's quite dry in many parts, or during some part of the year. And so the Phaseolus is definitely evolved in a different environment. So they again identify like two loci responsible for. And what is interesting, that one of them is complete homolog and ortholog in terms of the function with the soybean one. So you have a soybean in Asia, Phaseolus in America, very different environment, but there is a same gene responsible for pod dehiscence, which is a dirigent-like protein involved again in the lignification. So that's very nice, interesting story. I don't want to be late. There, of course, there is a recent another work from Roberto Papa lab, which show basically the transcription factor, which is likely, which is a MAP family transcription factor, which is involved in the regulation of this lignification of the pod, which is encoded by the second loci. I mentioned there are two in Phaseolus. There have been two in soybean, and this is the one. So one was a dirigent, and this is a MIP factor, which is very likely involved in the same pathway. So giving these examples, we have like kind of genes, and this is not the complete list, but we call them differently. So we have something called domestication category, which we found, let's say, necessary, or which we found like very highly selected for the domesticated crop. Then we have a category which is called like diversification, basically, which allowed to move to the different environment, like for example, shifting in geographic. So typically flowering time has been changed because we changed. Also some of the, let's say, the growth characters. So this is not necessary, but in some environment only. And then we have a category which is called improvement, which is typically, I put just for soybean, but we have it for any, which is like related to the specific human requirement. So it's relatively modern. So of course the resistances or some other shapes. I said the shattering in soybean is like kind of conditional, basically not necessary in some environment, but seed dormancy is definitely something very, very important. To the very end, actually in legumes we are sometimes jealous in terms of, because of one of the very fancy topics studied in plant sciences is the hybridization and polyploidy in terms of the speciation, for example. If you look at across legumes, this phenomenon is relatively very less frequent. I mean, it's infrequent. One of the example which is different is a peanut. Actually, the peanut has originated from the hybridization of two species in South America, and we know quite solid evidence that they have been, let's say, hybridized, and of course they have to be polyploid to be able to be fertile. What is the future? One of the illustration what the future might be is like this is the new hybrids of, let's say, distinct wild peanuts, which are crossable with a cultivated one, because normally they would be a barrier because of these different ploidy levels. But this is recent work of Bartioli lab showing that this is a way how we can go, because basically we can still do some let's say the crosses. And what I am kind of saying, and this was something which we done back during legato time, many of you have been participating, was like trying to, let's say, reverse the bottleneck and to use the progenitor in terms of the widening of the diversity. So I think there is still way to go. We still can do it quite systematically with current genomics in hands. And as I mentioned several times, there are questions if the same genes and pathway has been altered. I show you examples that we have one evidence now from Phaseolus and Glycine. I think it will be very exciting in a near future once we have more genomic and genetic data in hands. And the very end is sometimes people ask what it is good for. I think I don't need to really convince you, but for example, one friend of mine in Argentina, which is in the bottom part, is trying basically to domesticate the wedge, the hairy wedge, simply to remove the dormancy, to remove the podehiscence, and for that would be very useful to know the genes. So basically he is removing by selecting, but if we know the pathways and genes, we can do it much more efficiently. So that's one of my arguments why it's good to know the domestication. So I think it's good to know the history and to look at the future. So thank you for listening. I know they have been a little bit longer, but hopefully it was not too bad. Well, there are many people which are involved, and I really appreciate the collaboration with many. And thanks for listening. So please, and of course, yeah, I should acknowledge the P, so the Gregor Mendel and anniversary which we celebrated this year. So thanks a lot.
Thanks, Peter. It has been a wonderful webinar and very interesting. Now, if we have still a few minutes, if people have some question, it's a good time for you to ask.
Yeah, please.
I have a question. I don't know if I'm seen very good in this slide.
Yes, we are hearing you, Asa.
Okay, good. Thanks. Thank you for a very nice presentation. My name is Åsa Grimberg. I'm working at Swedish University of Agricultural Sciences. I'm working with faba bean, but I was very interested to hear about this, what you call it, increased permeability of water in the peas that you showed. So my question, I think, is if this trait has changed through the domestication process, did you mean that it has been connected to a human selective process, that they were looking for peas that were more beneficial in cooking and so on? Or did I misunderstood you there?
Well, you know, I think the most likely scenario was, of course, the germination. So to say, the aspect, if you place the seeds in the soil, you let them for the rain to come, how many of them would be germinating in terms of they not being dead, but they will be able to germinate, which is related to, of course, the imbibition and germination. I was saying that this is not proven, that another aspect could be, of course, the cooking ability. There are some arguments, for example, maybe even more in African legumes, that are kind of being able to imbibe quickly so you can cook them more rapidly. It's like one of the advantage. But I think the most likely scenario, the primary reason was the germination.
Okay, yeah, because I was also thinking of the anti-nutritional factors, if that could also withdrawn to some extent so that they survived better, so to speak.
Oh yeah, well, this is definitely connected to, you know, many of these what we call anti-nutrition, like trypsin inhibitors or whatever you can, convicillin or vicillin. Well, they have been removed by maybe the bitter taste or let's say something related to that. We know that now it's of course bad because we lost the protection. They could be also genetically linked, you know, because something which I slightly touch upon, some of these visual aspects, like less pigmentation or the hilum color, for example, we know some of these genes are closely, and it's less recombining, so to say. So basically they're like, you know, swapping together. So they might be not selected directly, but not indirectly in some way, right?
Thanks. Thank you.
There's another question from Paolo. Please speak up.
Yes,
Paolo.
thanks, Petr. Interesting indeed. As you mentioned, we are going to need more and more diversification, no? Especially to cope with climate change and the greater rising need for stress tolerance. But breeders, myself, I'm also a breeder. I mean, using land races, even more wild relatives, it is a real nightmare because of all these bad traits they carry. Now, what I dream of is having like a kind of array, you know, to select against, let's say, these unfavorable traits, so that just, you know, for key traits, I could easily, you know, from progenies select how far are we? I mean, if we wanted, how technically far are we in terms of knowledge? For example, for key crops such as pea, you work on, or soybean, would that be already feasible if we wanted? Do we have enough information for that?
Yeah, I know what you mean. Of course, first to your, let's say, nightmare of breeding. Yeah, that's definitely I'm aware of. I used to work with the breeders before. And yeah, once you do this cross, it takes you back because all of these or most of these traits are, of course, dominant. So basically, you ended up with a weed-like or wild-like material. And that's something which has to be done, as you know, systematically. We still haven't finished, basically. So you should do the repeated back crosses, and once you make these lines, they should be, let's say, used in breeding. So there is still, it typically takes a long time. How far we are? That's the right question. Well, I think the knowledge is still little. It's quite fragmented. I should say. At least my feeling is. We know relatively few of the let's say key genes, or let's say genes which were really interested. We know, of course, some flowering time genes because of the pathway is well conserved. We know few examples which I showed, but this might not be the necessary ones. But otherwise, there is a plethora of traits which we have no idea still. I think it's still, I mean, genetic. I don't know if you have asked, if you do the cross, how quickly you can select for something desirable, right? That was maybe your kind of scenario.
Definitely.
I don't think, irrespective of how many associations and other mapping has been done, it's basically mostly like genotype specific. I don't think that we are kind of very broad. I think except of flowering, and maybe someone can correct me, flowering, maybe some seed composition traits, but not many. I'm not aware of many good ones. They're like not general. I don't think that we have still enough information. I'm not aware how much is done. I suspect that soybean in terms of what the companies has been investing will be likely far more studied and without publishing, of course. So I don't know. But my feeling, what I know in, let's say, in our field or our trades, we still, it's kind of fragmented. There is no general picture to me.
Okay. Are there other questions? Yes, Akiko. Akiko, if you are talking, you are muted and we are not hearing you.
Hello?
Okay.
Hello?
Now.
Can you hear? Thank you very much. I cannot turn on the video, so I'm sorry. Thank you very much for very interesting talk. And I'm interested to know how this domestication event affected plant resistance to pests or pathogens. Do you know any studies?
Well, that's not, well, I mean, it is study, of course. We know that many of the, that's the long, let's say, the field of trying to find the resistant genotypes. But if you look at also globally, well, there are some resistances present, of course, in a domesticated material. But I think there is a good evidence for quite some time already that that there is a much wider repertoire of the resistance genes and alleles, so to say, of the genes in the progenitors. So I think that that's one of the arguments why we should try to look at them. Maybe more specifically, basically, if you know the susceptibility gene, you should try to look at and that's easier for crossing, of course. If you are crossing something which you know, which you are selecting for, that could be good. But what is, I think, very intriguing also and far less studied is the relationship to the microbiome, or to say, I mean, in terms of the relationship of the let's say beneficial microflora, for example. I think we neglected this over the domestication because basically we favor everything around the plants, so they didn't need to care about so much of their nutrition. I think generally speaking, the root system is something which has been not very much, at least recently, basically was not selected for. So I think that's quite interesting. From me, it's quite interesting for someone to look at.
Thank you very much.
Okay, are there any other questions for Dr. Smykal? Yes, Juan.
Yes, thank you, Peter, for your presentation. I've been a fan of your work for across many years. I've been following all your work, so that's great. It's always nice when someone else cites your work. I'm happy to see that you show some of our work on seed dormancy on common beans, so that was great. Yeah. We have some preliminary data that shows also that seed inhibition is related to flavonoid compounds and the flavonoid profile. Do you have any comments about that?
Yeah, I agree. Yeah, well, that's actually, I haven't shown any metabolite profiling. We are doing this extensively. It's not only, you know, it's a question, okay, what do you call flavonoids? I mean, they may be, I think the tricky part of all these metabolic analysis, and I'm doing this over last 10 years, is that you typically like, and I'm not analytical chemist, but the colleagues are, you are, let's say, extracting and sampling subset, right? Because you can imagine then a cell wall, it's quite complex. You have a flavonoid and phenolic compounds, maybe or very likely linked to the hemicellulose and linked to the other compounds like fatty acids and so it's a kind of complex mixture. But I agree with you, and I would be happy to share with some of our preliminary, there is a relationship, definitely. Well, it was shown in Arabidopsis because even in Arabidopsis, the seed coat permeability plays some role, and it has been known that there is a flavonoid series mutants, which, as you are aware of this. So I agree, yeah, there is a relation. But that's also could be related also to this fact of the removal of a pigmentation. You know, I was mentioning provocatively, but it's not a general rule. I think the common bin which you are working on is one of the good examples that it's not the case, because you can have perfectly
We have black beads, yeah.
Black beads. Yeah, sure. I'm aware of that. But there is some differences. Yeah, I agree.
Okay. I'll contact you later. And
yeah, definitely.
I think we have interesting data regarding flavonoids that I would love to share with you. Yeah.
Yeah. I can share with you as well ours. Great.
Okay. Thanks. That there's a lively public. Are there any other more questions? Okay, so I guess that is time for us to wrap up. I thanks again Petr Smykal for his nice contribution to our webinars, and I say again, this has been a webinar for the International Legumes Society. We have recorded everything. Probably today I'm going to upload everything, and you can find all the past recording on our website. And I leave you with a reminder that next month we'll have on November 8th another webinar from Julia Witnick from IRE. And next month we are going to talk about seed vigor in legume, how to improve it. So thanks, and see you next month.
Thanks for everyone. Yeah, it was great. So
Bye.
take care. Bye, everyone.